Humans use a lot of energy, especially through their use of transportation such as cars. The problem with cars is that they mainly run on fossil fuel, a source of hydrocarbon that takes billions of years to replenish itself. A great alternative for fossil fuel could be biofuels, which are fuels made from biomass, plants, or small organisms that store the sun's energy through photosynthesis. The great thing about biofuel is that the carbon dioxide released when it's burned will then be reused by the same plants or algae when they were once again grown to produce biofuel, inferring that it is carbon neutral, which is not the case of fossil fuel as they release tons of CO2 that used to be trapped in the earth when burned. And so this project will be about biodiesel, a type of fuel that is made from algae. March 13, 2018, 1637. Today was the first day of the lab of the biodiesel experiment. We, in order to collect oil from algae, have prepared a solution of BBM, glycerol, and chlorella vulgaris in order to grow our own supply of algae. As you may see, they are now resting upon some magnetic tables and will have to come once or twice a week to take some spectrometry data in order to monitor its growth. March 28, 2018, 13:50. Hello, so today I'm going to take my spectrometer measurement to monitor the growth of the alga. And since we're talking about alga, I brought a sushi sheet for scale to compare them. Now, all joke apart, I'm actually going to take some spectrometer measurement in order to monitor the growth of the alga according to how much of the light it filters. And so, the more the light it filters, the more it has grown. Now, let's just see how it goes. So today we've done the second step of the biodiesel lab, which was just keeping track of the progress of the growth of the alga, as well as the impact of glycerol on its growth. And so we use spectrometry for that. Now, I first started by calibrating the spectrometer with the according basic solution used for each alga, which was F2 for Nanochloropsis and BBM for Chlorella vulgaris. I then proceeded to take the measurement of the growth of the alga without glycerol, which was named control, and the one with glycerol, and the results were drastically different. And so the algae with glycerol gave a measurement of 0.625, as the one without glycerol gave a measurement of 0.151. That glycerol helped the growth of the alga, as the one with glycerol blocked more of the light than the one without, as revealed by the measurement. And so I was quite surprised by how different the measurement revealed to be, as it was nearly 4 times bigger for the one with glycerol than the one without. March 3rd, 2018, 12:03. Today was the third part of the biodiesel project, which had as a purpose to observe algae with a normal microscope to monitor its growth with an hemocytometer as seen here. An hemocytometer is separated into 4 big squares, which are themselves separated into 16 smaller ones. It is used to try and estimate the number of cells per ml of a solution for an algae using this equation. We also compare yeast and our algae in order to clearly determine that algae does photosynthesis, explaining their green color and the presence of chloroplast, as yeast did not. After successfully determining that algae made photosynthesis and had grown until a certain point, we observed celled algae under a fluorescent microscope to observe other organelles under a different light, literally. And so, after adding a special chemical that would stain the oil contained by the algae, it was possible to observe bright red spot of color within the algae cell's wall in what we call oil droplets. March 3rd, 2018, 16:28. The fourth part of the biodiesel experiment consisted of taking another spectrometer measurement in order to, using diagram, actually calculate the number of cells per ml of our algae solution, which revealed to be 1,905,000, as our spectrometer measurement gave an average value of 1.223. Our algae solution was then centrifuged at 6,000 rpm several times to separate the actual algae from the BBM solution. We then proceeded to separate, rehydrate, and heat up our algae until it turns to a crisp so that we could manually crush it to extract the oil. A lot of different methods could have been used, such as adding supercritical fluid to the algae, but manually crushing them remains the simplest and easiest way of extracting the oil. To conclude, I was quite surprised to realize that our spectrometer measurement was over the value of 1, as I thought the result of a spectrometer measurement was a ratio of how much of a certain wavelength, in this case 685 nanometer, which is red as it corresponds to the peak of absorbance of the chlorophyll, is absorbed. However, I was shown the actual equation and it revealed to be the natural algorithm of said ratio, which explains why a value over 1 and a value below 1 is plausible. March 5th, 2018, 1742. Today was the fifth part of the Biodiesel experiment where we try and determine the concentration of free fatty acid of different oil through titration. It has to be noted that we couldn't use our algae because the quantity that we produced was far from sufficient to have an actual sample. The titration was done in order to find the exact value of KOH that we needed to add to each type of oil in order to just neutralize it, as adding any more than that during the trace esterification, a step in which the oil is transferred to biodiesel, would result in the creation of soap, which can be the separation of the biodiesel from the glycerol at the end. The final value ended up being 0.29 gram for soybean oil and 0.40 gram for used oil, which makes sense as the concentration of FFA for used oil was higher than the one of soybean oil, 0.18% compared to 0.05%. I had never done any titration before and was quite surprised of how much a single drop of titrant could completely tilt over the balance of the solution, rendering the trial useless as the solution would certainly go from being basic to acid without an intermediate state. March 6, 2018, 10:08. Today was the sixth part of the biodiesel experiment where we finally produced biodiesel from oil. In order to do so, we mixed the oil with methanol and KOH and stirred them over some magnetic plate until the reaction reached its equilibrium and biodiesel was formed. Even at this point, it was possible to determine that the biodiesel, after the transesterification, the name of a process in which an oil is converted to biodiesel, would be floating on top of the glycerol as it has a lesser density. It was also deemed really important to keep the reaction in between a temperature of 45 and 55 degrees Celsius, as, even though the reaction is held by heat, the boiling point of methanol is quite low, 65 degrees Celsius, and so it is of our best interest to make sure that one of our reactants does not evaporate, as it would change its concentration and inevitably influence the reaction through Le Chatelier's principle. And so in the end, I was pleasantly surprised of how simple the transesterification process was, as I thought creating biodiesel was a way more complex and intricate thing to do. March 9, 2018, 10:13. Today was the seventh part of the biodiesel project in which we proceeded to refine our biodiesel. To do so, we decanted the solution, a simple but difficult process of pouring the biodiesel into a new receptacle without contaminating it with the glycerol underneath. After rinsing it with water to remove some of the impurities such as soap or leftover of the transesterification process, our final volume of biodiesel revealed to be 54.5 ml for the soybean oil and 53 ml for the used oil, which gives us a percent yield of 94.35 for the virgin soybean oil. Accordingly, when comparing the percent conversion of volume of the two oils, it was observed that the percent conversion of the soybean oil was higher than the one of the used oil, 99.09% compared to 96.36. This can be explained by the higher initial concentration of free fatty acid in the used oil, as these FFA tends to react with the catalysis, the KOH, to yield soap and not biodiesel, thus wasting some precious fatty acids that could have been converted to biodiesel. Now, although the process of making biodiesel wasn't as hard as I thought it would be, it was still a long and exhaustive procedure that took several weeks. This is why we came up with the idea of an automatic machine that could do the process for us. March 12, 2018, 17:15. Today was the eighth part of the biodiesel experiment where we burned different type of fuel to compare the efficiency. In order to do so, we heated a set amount of water using different fuels and calculated the number of joule emitted and received compared to the mass of combustible burnt. However, our results were inconclusive as our biodiesel would technically have to be the one with the highest heat of combustion because it is the molecule which contains the most energy. This can be explained by the remaining impurities in our biodiesel that was only watched once. In the end, I was quite surprised by the smell exhaled by the used oil, which was almost aromatic and stood out among the others. March 13, 2018, 11. Today was the final part of the biodiesel experiment where we were shown an almost completely automatic biodiesel machine. This ingenious device utilizes gravity and automatic valves that are timed so that different reactant used in the transit purification and washing process of producing biodiesel will only mix and react in a certain order and in a particular container. However, the very last step of collecting the biodiesel must still be done by hand.